Happy morning, dear students. Welcoming you all to this online class. So, in previous class, we completed immunology lesson. Today, we start the new chapter, microbes in human welfare. In this lesson, we are going to discuss some of the microbes in household products, microbes in industrial products, etc. Microbes. Microorganisms such as bacteria, fungi, protozoa, certain algae, viruses, and prions are some of the major components of biological system on Earth. Several microorganisms are beneficial and contribute to human welfare. Microbes are present everywhere in soil, water, air, and within the bodies of plants and animals. Microbes like bacteria and fungi can be grown on nutritive media to form colonies which can be visibly seen. We will discuss some of the microbes in household products. In everyday life, microbes and their products are used in the preparation of idli, dosa, cheese, curd, yogurt, duff, bread, vinegar, etc. Bacteria like Lactobacillus acidophilus, Lactobacillus lactis, and Streptococcus lactis, commonly called as lactic acid bacteria. Lab, simply we can call it as lab bacteria. So these are probiotics. Probiotic means which is useful, which check the growth of pathogenic microbes in the stomach and other parts of the digestive tract. The lab bacteria grows in milk and convert it into curd, thereby digesting the milk protein casein. A small amount of curd added to fresh milk as a starter or inoculum contains millions of lactobacilli which under suitable temperature, that means below 40 degrees centigrade, multiply and convert milk into curd. ஒன்றும் இல்லை, பாலில் கொஞ்சம் தயிர் ஊற்றி வச்சோன்னா என்ன ஆகும், அது மறுபடியும் கேர்டாக மாறுது. Curd is more nutritious than milk as it contains a number of organic acids and vitamins. First we see what is probiotic and prebiotics. Prebiotics are compounds in food, that means the fibers that induce the growth of beneficial microorganisms. Prebiotic means induce the growth of beneficial microorganisms. Probiotics means the microorganisms intended to provide health benefits when consumed. அதை சாப்பிட்டோன்னா நம்மளுக்கு health benefit இருக்கும். That is called as probiotics. Yogurt. Yogurt is produced by bacterial fermentation of milk and lactic acid is produced as a byproduct. Microorganisms such as Streptococcus thermophilus and Lactobacillus bulgaris coagulate the milk protein and convert the lactose in the milk to lactic acid. The flavor in yogurt is due to acetaldehyde. It's a 1-mark question. So yogurt அந்த தயிர் வாசனை கொஞ்சம் நல்லா இருக்கும், அந்த காரணம் acetaldehyde. Cheese is a dairy product produced in a wide range of flavors, textures, and is formed by coagulation of milk protein casein. So casein நால உருவாக்கப்படுறது தான் cheese. During cheese production, milk is usually acidified and the enzymes rennet is added to cause coagulation. So அந்த பாலை திரிய வைக்கிறதுக்காக enzyme rennet add பண்றாங்க. The solids are separated and pressed to form cheese. Most cheese made with a starter bacteria, Lactococcus, Lactobacillus or Streptococcus. Next is paneer. Paneer is a cottage cheese. It's a fresh cheese common in South Asia, especially in India. It is made by curdling milk with lime juice. Lime juice add பண்ணா அந்த பால் திரிஞ்சிடும். Not only lime juice, vinegar and other edible acids. Large holes in the Swiss cheese is due to the production of large amount of carbon dioxide by the bacterium Propionibacterium shermani. ஓகே, ஸோ அந்த ஸ்விஸ் சீஸில் பார்த்தோன்னா டாம் அண்ட் ஜெர்ரிலாம் பார்த்தீங்கன்னா தெரியும், நடுவில் பெரிய பெரிய ஓட்டெல்லாம் இருக்கும். ஸோ அந்த சீஸில் இருக்கக்கூடிய அந்த ஹோல்ஸ்க்கு காரணம் என்ன அப்படின்னு பார்த்தோன்னா due to the bacterium Propionibacterium shermani. The duff used in the preparation of duffna maavu. The duff used in the preparation of idlis and dosas are fermented by the bacteria Leuconostochum mesenteroides, whereas the duff used in the bread making பிரெட் யூஸ் பண்ணுறதுக்கான அந்த மாவு அதை ஃபர்மெண்ட் பண்ணுறது எதுன்னு பார்த்தோன்னா சாக்ரோமைசிஸ் cerevisiae, that is Bacchus yeast. Fermentation of glucose mainly forms ethyl alcohol and carbon dioxide, which is responsible for leavening of dough. அது லேயராக வரும், அந்த ஒரு ஓட்டெலாம் நடுவில் வருது இல்லையா, அதுக்கு காரணம் என்ன அப்படின்னு பார்த்தோன்னா பிகாஸ் ஆஃப் திஸ் கார்பன் டை ஆக்சைடு. When leavened dough is baked, both carbon dioxide and ethyl alcohol evaporate, making the bread porous and soft. Next is the single cell protein, SCP. What is single cell protein? Single cell protein refers to edible unicellular microorganisms like spirulina. It is a single cell protein, that means edible சாப்பிடக்கூடிய unicellular microorganism. Protein extracts from the pure or the mixed cultures of algae, yeast, fungi or bacteria may be used as an ingredient or a substitute for protein-rich foods and is suitable for human consumption or as animal feed. So this single cell protein is used as an ingredient or substitute for a protein. Next we move on to the microbes in industrial products. Microbes are used to synthesize number of products, valuable products to human beings. So first is the antibiotic production. Antibiotics are chemical substances produced by microorganisms which can kill or retard the growth of other diseases causing microbes even in low concentration. ஸோ ஒரு மைக்ரோ ஆர்கானிசமை யூஸ் பண்ணி இன்னொரு மைக்ரோ ஆர்கானிசமை சாகடிக்கிறது, தட் இஸ் கால்ட் ஆஸ் ஆன்டிபயோட்டிக். Antibiotic means against life. Antibiotics are used to treat diseases such as plague, meningitis, diphtheria, syphilis, leprosy, tuberculosis, etc. Silvan Waxman discovered the streptomycin and was the first to use the term antibiotic. So the term antibiotic was first used by Silvan Waxman. So he only discovered streptomycin in the year 1943. While working on Staphylococci bacteria, Alexander Fleming observed a green mold growing in one of his unwashed culture plates around which Staphylococci could not grow. So Staphylococci bacteriaவை வச்சு ஏதோ ஒரு எக்ஸ்பெரிமெண்ட் பண்ணிட்டு இருக்கும்போது அலெக்சாண்டர் ஃப்ளெமிங் அப்சர்வ் பண்ணார். ஸோ அன்வாஷ்டு பிளேட்ஸில் பார்த்தோன்னா அது ஒரு க்ரீன் மவுல்ஸ் உருவானதை பார்த்தார். He found that it was due to the chemical produced by the mold and he named it as penicillin, which was the first antibiotic discovered by Alexander Fleming in the year 1926. Penicillin is produced by the fungi Penicillium notatum and Penicillium chrysogenum. It is a bactericidal. Bactericidal means the antibiotic which kills the bacteria in action and inhibits the synthesis of bacterial cell wall. Penicillin is also referred to as queen of drugs, and its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Florey when they treated the wounded soldier in World War II with penicillin. Penicillin is called as queen of drugs. ஸோ அதனோட எஃபெக்ட் எப்போ தெரிஞ்சுதுன்னு பார்த்தோன்னா செகண்ட் வேர்ல்டு வார் அப்போ ஏர்னஸ்ட் செயின் அண்ட் ஹோவர்ட் ஃப்ளோரி இவங்க என்ன பண்ணாங்க, wounded சோல்ஜர்ஸ்க்கு இந்த பெனிசிலின் அப்ளை கொடுத்தாங்க. ஸோ அது வந்து நல்லா ஒர்க் அவுட் ஆச்சு. ஸோ ஃப்ளெமிங், செயின் அண்ட் ஃப்ளோரி were awarded the Nobel Prize in the year 1945 for the discovery of penicillin. Antibiosis. What is antibiosis? It is a property of antibiotics to kill the microorganisms. Antibiotics is of two types. One is broad spectrum antibiotic, narrow spectrum antibiotic. Broad, it acts against a wide range of diseases causing bacteria. Narrow means these are active against selected group of bacterial types. Selected ஒரு சில இதை மட்டும்தான் அழிக்கும், அது narrow. எல்லாத்தையும் அழிக்கும், அதுக்கு பேர் broad. Right? Next is the tetracycline. Tetracycline is a broad spectrum bacteriostatic antibiotic. Bactericidalனா பாக்டீரியாவை கில் பண்ணும். Bacteriostatic அப்படின்னா ஸ்டாட்டிக்னா அப்படி நிறுத்துறது. It stops the growth of the bacteria that inhibits microbial protein synthesis. Chlorotetracycline is the first antibiotic of this group, isolated from the cultures of Streptomyces aureophasian. Streptomycin is a broad-spectrum antibiotic isolated from the actinomycetes. Streptomyces gracious. It is a bactericidal against both gram-positive and gram-negative bacteria, especially against Mycobacterium tuberculosis. Streptomycin work against the Mycobacterium tuberculosis. Antibiotics such as erythromycin, chloromycetin, grisufluin, neomycin, kanamycin, bezitrazen, etc. are also isolated as microbial products. So what is hypersensitivity reaction? It is a major problem with the use of penicillin. பெனிசிலின்லாம் இப்போ நம்ம பெனிசிலின் இன்ஜெக்ஷன் போடுறதுக்கும் போது பார்த்தோன்னா முதல்ல ஒரு டெஸ்ட் ஊசி போடுவாங்க இல்லையா, கையில் ஏதாவது கொஞ்சோண்டு மருந்து செலுத்தி நம்மளுக்கு ஏதாவது இன்ஃபெக்ஷன் ஆகுதா, ஸ்கின்ல ரேஷஸ் ஏற்படுதா அப்படின்னு பார்த்துட்டு தான் நம்மளுக்கு வந்து பெனிசிலினை யூஸ் பண்ணுவாங்க. ஸோ ஹைப்பர் சென்சிட்டிவிட்டி ரியாக்ஷன் இஸ் த மேஜர் ப்ராப்ளம் வித் த யூஸ் ஆஃப் பெனிசிலின், ரிசல்ட்டிங் வித் nausea, vomiting, wheezing, and ultimately cardiovascular collapse. To check the sensitivity reactions, doctor use a needle to prick the forearm of the patient to give a weak dose of penicillin. And each red region in the forearm is an indication that patient is allergic to penicillin. அவங்க போட்ட ஊசி அப்படி போட்டுட்டு ஒரு 1/2 hour விட்டுருவாங்க. இந்த இடத்துல எந்த அலர்ஜியும் ஏற்படலை அப்படின்னா நம்மளுக்கு மறுபடியும் அந்த மருந்து கொடுப்பாங்க. ஏதாவது ரேஷஸ் மாதிரி சிகப்பு கலரில் அப்படியே அந்த இடம் ஸ்கின் மாறிச்சுன்னா நம்மளுக்கு அதை கொடுக்க மாட்டாங்க. ஓகேவா? ஸோ ஹைப்பர் சென்சிட்டிவிட்டி ரியாக்ஷன் இஸ் மேஜர் ப்ராப்ளம் வித் த யூஸ் ஆஃப் பெனிசிலின். நெக்ஸ்ட் இஸ் antibiotic resistance. Antibiotic resistance occurs when bacteria develops the ability to defeat the drug designed to kill or inhibit their growth. Antibiotic resistance. So it is one of the most acute threat to public health. Antibiotic resistance is accelerated by the misuse and overuse of antibiotics, as well as poor infection prevention control. Antibiotics should be used only when prescribed by the certified health professional. யார் வேணாலும் ஆன்டிபயோட்டிக் எடுக்கக்கூடாது. டாக்டர்ஸ் என்ன ப்ரெஸ்கிரைப் பண்ணுறாங்களோ அதை தான் நம்ம யூஸ் பண்ணணும். டூ மச்சாகவும் எடுக்கக்கூடாது, ரொம்ப கம்மியாகவும் இருக்கக்கூடாது. When the bacteria become resistant, antibiotics cannot fight against them and the bacteria multiply. Narrow spectrum antibiotics are preferred over the broad spectrum antibiotics. They effectively and accurately target specific pathogenic organisms and are less likely to cause resistance. Superbug is a term used to describe the strains of bacteria that are resistant to the majority of antibiotics commonly used today. So what is a superbug? Superbug is nothing but genetically modified bacteria is called as superbugs. Right? Next, we want to fermented beverages. Microbes, especially yeast, is being used from time immemorial for the production of beverages like wine, beer, whiskey, brandy, and rum. Wine is among the oldest alcoholic beverages known and is produced by the fermentation of fruit juice by yeast. So wine fruit juice fermentation பண்றதுனால கிடைக்கக்கூடியது. Zymology is an applied science which deals with the biochemical process of fermentation and its practical uses. Zymology, zyme means enzyme. It deals with the biochemical process of fermentation. Saccharomyces cerevisiae, commonly called brewer's yeast, is used for fermenting malted cereals and fruit juices to produce various alcoholic beverages. So yeast is used for fermenting cereals and fruit juices. Wine and beer are produced without distillation, whereas whiskey, brandy, and rum are obtained by fermentation and distillation. Wineum beerum பார்த்தனா distillation வடிகட்ட மாட்டாங்க, whereas whiskey, brandy, rum இதெல்லாம் fermentation பண்ணி distillation பண்ணுவாங்க. So the Pasteur effect is the inhibiting effect of oxygen on the fermentation process. What is enology? Enology is the science and study of wine and winemaking. Wine is made from the fermentation of grape juice. Grape juice is fermented by various strains of Saccharomyces cerevisiae, that means yeast, into alcohol. Grape wine is of two types, red wine and white wine. For red wine, black grapes are used, including the skins, and sometimes the stems are also used. In contrast, white wine is produced only from the juices of either white or red grapes without the skin and stems. Beer. Beer is produced from germinated barley malt grains by Saccharomyces carbagens or Saccharomyces cerevisiae. Rum is made from the fermented sugarcane or molasses or directly from sugarcane juice by Saccharomyces cerevisiae. பீர்ன்றது பார்லி அதை ஜெர்மினேட் பண்ணி அதுவும் ஒரு Saccharomyces cerevisiae யூஸ் பண்ணுறாங்க. ரம்மும் it is a fermented sugarcane. ஓகேவா, the molasses that is produced from the sugarcane. Okay, whiskey is a type of distilled alcoholic beverage made from the fermented grain mash by Saccharomyces cerevisiae. So yeast is the major part that is produced in the production of this alcoholic beverages. In some part of South India, a traditional drink called padanir is obtained from fermenting sap of palms and coconut trees. தென்னை மரம் பனைமரத்திலிருந்து எடுப்பாங்க இல்லையா பதநீர். A common source is tapping of unopened spadices of coconut. It is a refreshing drink which on boiling produces jaggery. இது ஒரு refreshing drink. இதை boil பண்ணால் jaggery or palm sugar பனவெல்லம் எடுக்கலாம். So when padanir is left undisturbed for a few hours, it gets fermented to form toddy, kallu, with the help of naturally occurring yeast to form a beverage that contains 4% alcohol. After 24 hours, toddy becomes unpalatable. அப்புறம் கொஞ்ச நேரம் அந்த மறுபடியும் வச்சுருந்தோன்னா அந்த கல் கூட நம்ம என்ன பண்ண முடியாது, சாப்பிட முடியாது. ஓகே, and it is used for the production of vinegar. Saccharomyces cerevisiae is the major producer of ethanol. Saccharomyces yeast is the major production or producer of ethanol. It is used for industrial, laboratory and fuel purposes. So, ethanol is referred as industrial alcohol. Bacteria such as Zymomonas mobilis, Sarcina ventriculi are involved in ethanol production. The principal substrates for the commercial production of industrial alcohol include molasses or corn, potatoes and wood waste. The process of ethanol production starts by milling a feedstock followed by the addition of dilute or fungal amylase. It is an enzyme from Aspergillus to break down the starch into fermentable sugars. Yeast is then added to convert the sugar to ethanol, which is then distilled off to obtain ethanol, which is up to 96% in concentration. The two common types of biofuels in use today are ethanol and biodiesel. Both of them represent the first generation of biofuel technology. Ethanol is often used as a fuel, mainly as a biofuel additive for gasoline. Next, biofuel. Biofuel is a fuel used from the vegetable oils, fats, or greases. Biofuels can be used in diesel engines without altering the engine. Pure biodiesel is a non-toxic, biodegradable and produces a lower level of air pollutants than the petroleum-based diesel fuel. The government of India approved the national policy on biofuels in December 2009 and identified Jatropha curcas as most suitable oil seed for biodiesel production. Pongamia species is also suitable choice for the production of biodiesel. How this ethanol is produced? C6H12O6, that is glucose, on fermentation by yeast gives C2H5OH plus carbon dioxide is released. World Biofuel Day is observed every year on 10th August to create awareness about the importance of renewable biofuels as an alternative to conventional non-renewable fossil fuels. This day also highlights the various efforts taken by the government in the biofuel sector. Next, what are the chemicals, enzymes and other bioactive molecules used by these microbes? Microbes are not only used for commercial and industrial production of alcohol, but also used for the production of chemicals like organic acids and enzymes. Examples of organic acid producers are Aspergillus niger for citric acid, Acetobacter aceti for acetic acid, Rhizopus oryzae for fumaric acid, Clostridium butyricium for butyric acid, and Lactobacillus for lactic acid. So citric acid, acetic acid, fumaric acid, butyric acid, and lactic acid can be produced from these microbes. Yeast, Saccharomyces cerevisiae, and bacteria are used for commercial production of enzymes. Enzymes such as lipases are used in detergent formulations and used for removing oily stains from laundry. The oil கரையெல்லாம் போக்குறதுக்கு lipases. Bottle juices are clarified now purified by the use of pectinase, protease, and cellulase. Rennet can also be used to separate milk from solid curds for cheese making. Streptokinase produced by the bacterium Streptococcus and genetically engineered streptococci are used as clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction. Myocardial infarction is nothing but heart attack. இப்போ ஹார்ட்டில் ஏதாவது பிளாக் ஏற்பட்டால் ஓகேவா இப்போ ஏதாவது பிளாக் ஏற்படுதுன்னா அதை genetically engineered streptococci which is used as a clot buster. With help of this we can able to clear the clots, blood clot inside the blood vessels. blood clot ஆனால் தான் என்ன ஆகும் ஹார்ட் அட்டாக் வர்றதுக்கான வாய்ப்புகள் இருக்குது. அந்த ஹார்ட் அட்டாக் வராமல் தடுக்கணும் அப்படின்னா இந்த clot buster நம்ம யூஸ் பண்ணலாம். Right? Cyclosporin A, an immunosuppressant used in organ transplantation, is produced from the fungus Trichoderma polysporum. Cyclosporin அப்படின்ற ஒரு immunosuppressant, that means it suppresses the immune system during organ transplantation. It is used for its anti-inflammatory, antifungal and anti-parasitic properties. Anti-inflammation வீக்கத்தை தடுக்கும், ஃபங்கல் இன்ஃபெக்ஷன் வராமல் தடுக்கும், parasitic இன்ஃபெக்ஷன் வராமல் தடுக்கும். அடுத்தது statins. Statins produced by the yeast Monascus purpuris have been used to lower the blood cholesterol levels. So the yeast such as Monascus purpuris have been used to lower the blood cholesterol levels. It acts by competitively inhibiting the enzymes responsible for the synthesis of cholesterol. Recombinant human insulin have been produced predominantly using this E. coli, that means Escherichia coli and Saccharomyces cerevisiae for therapeutic use in human. So human recombinant human insulin, it can be also produced with help of this yeast and E. coli. Sewage treatment and energy generation. What is a sewage? Sewage is the waste generated every day in cities and towns containing human excreta. It contains large amount of organic acid and microbes which are pathogenic to humans and are biodegradable pollutants. Domestic waste consists of approximately 99% of water, suspended solids, and other soluble organic and inorganic substances. Sewage should not be discharged directly into the natural water bodies like rivers and streams. Before disposal, sewage should be treated in sewage treatment plants to make it less polluting. So this sewage treatment includes 3 different stages: the primary treatment, secondary treatment, and tertiary treatment. In primary treatment, the suspended and the floating materials are removed. And then in secondary treatment, this water is combined with the microorganisms. These microorganisms engulf the other microorganisms present in water. And the tertiary treatment includes addition of chemicals or using of ultraviolet rays to clean up this sewage water. This we will discuss more detail in chapter 13. And then we move on to microbial fuel cell, MFC, microbial fuel cell. Microbial fuel cell is a bioelectrochemical system that drives an electric current by using bacteria and mimicking the bacterial interaction found in nature. So with the help of the bacteria, this microbial fuel cell is constructed. Microbial fuel cells work by allowing bacteria to oxidize and reduce organic molecules. Bacterial respiration is basically one big redox reaction. You know that what is meant by redox reaction? Redox means reduction and oxidation reaction in which electrons are being moved around. A microbial fuel cell consists of an anode and a cathode separated by a proton exchange membrane. In this figure, you can able to see an anode and cathode which is separated by a proton exchange membrane. Microbes at the anode oxidize the organic fuel generating protons which pass through the membrane to the cathode, and the electrons pass through the anode to the external circuit to generate current. See, நிறையா நம்ம செல்ஸ் பேட்டரிஸ் பார்த்துருப்போம். ஸோ இது வந்து by using these microbes we can create a electric circuit. Okay, and next one is microbes in the production of biogas. Biogas is a mixture of different gases produced by the breakdown of organic matter in the absence of oxygen. Biogas can be produced from raw materials such as agricultural waste, manure, and then municipal waste, plant material, sewage, food waste, etc. Biogas is produced under anaerobic condition. Anaerobic means without the use of oxygen. That means in the absence of oxygen. When the organic materials are converted through the microbiological reactions into gas and organic fertilizer, biogas primarily consists of methane. One mark. Primarily consists of methane. About 63% of methane is present in biogas along with carbon dioxide and hydrogen. Methane producing bacteria are called as methanogens, and one such common bacterium is methanobacterium. The methane producing bacteria are called as methanogens. Biogas is devoid of smell and burns with a blue flame without smoke. The methanogens are also present in anaerobic sludge and the rumen of cattle. Rumen means water inside the digestive tract of the cattle. So in rumen, these bacteria help in the breakdown of cellulose. Cellulose is present in plants. Okay, so to digest this cellulose, bacteria helps in some of the cattle. The excrete of cattle called dung is commonly called as gobar. The excrete of cattle is called as gobar. Gobar gas is generated by the anaerobic decomposition of the cattle dung. It consists of methane, carbon dioxide with some hydrogen, nitrogen and other gases in trace amounts. In biogas plant, anaerobic digestion is carried out in an airtight cylindrical tank known as digester. You can see in this picture. A digester is present. This is the biogas plant in which there is a cylindrical tank known as digester, and it is made up of concrete bricks and cement or steel. Bio waste are collected and slurry of dung is fed into this digester. This had a side opening into which the organic materials for digestion are incorporated for microbial activity. Anaerobic digestion is accomplished in 3 stages. One is solubilization, next one is acidogenesis, and third one is methanogenesis. The outlet is connected to a pipe to supply the biogas. The slurry is dried through another outlet and is used as fertilizer. Biogas is used for cooking and lighting. The technology of biogas production was developed in India mainly due to the effort of Indian Agricultural Research Institute IARI and Khadi and Village Industries. That's all about the biogas plant. You can able to see this biogas plant in villages. Okay, wow. அப்போலாம் ஒரு 10 to 15 years or 20 years back. ஸோ இந்த மாதிரி கேஸ் ப்ரொடக்ஷன் இருந்துச்சு இன் வில்லேஜஸ். அந்த கவுடங்லாம் வேஸ்ட் பண்ண மாட்டாங்க. ஸோ வீட்டில் இருக்கக்கூடிய முனிசிபல் வேஸ்ட் அண்ட் கவுடங் இது எல்லாமே இந்த டைஜஸ்டரில் கொட்டி அதிலிருந்து பயோகேஸ் உற்பத்தி பண்ணி அவங்க என்ன பண்ணாங்க, இட் இஸ் யூஸ்டு ஃபார் குக்கிங். ஓகே, நெக்ஸ்ட் ஒன் இஸ் bioremediation. What is meant by bioremediation? The use of naturally occurring or genetically engineered microorganisms to reduce or degrade pollutant is called bioremediation. It's an important 2-mark question. The use of naturally occurring genetically engineered microorganisms. That means modification is made in the microorganisms to reduce or to degrade the pollutants, that is called as bioremediation. Bioremediation is less expensive and more sustainable than other remediations available. It is grouped into in situ remediation, that means treatment of a contaminated soil or water in the site, or ex situ bioremediation, that means treatment of contaminated soil or water that is removed from the site and treated. In situ na அந்த இடத்துல வச்சு செய்யறது, ex situ அப்படின்னா அங்கேருந்து எடுத்துட்டு போய் வேற இடத்துல வச்சு செய்யறது, அவ்வளோதான். Microorganisms involved in bioremediation. Aerobic microbes degrade the pollutants in the presence of oxygen. Aerobic means in the presence of oxygen. They mainly degrade pesticides and hydrocarbons. Pseudomonas putida is a genetically engineered microorganism. The Pseudomonas putida is called as superbug. Okay, superbug is nothing but genetically engineered microorganism. Pseudomonas putida is a superbug which was first obtained by Anand Mohan Chakravarti by the recombinant bacterial strain. It is a multiplasmid hydrocarbon degrading bacterium which can digest the hydrocarbons in the oil spills. So seas like shipலாம் இடிச்சிக்கிச்சுன்னா ஓகேவா, ஸோ ஆயில் ஸ்பில் ஆயிடுச்சுன்னா திஸ் gem, that means Pseudomonas putida, is used to digest the hydrocarbons. And then Nitrosomonas europaea is also capable of degrading benzene and variety of halogenated organic compounds including trichloroethylene and vinyl chloride. Nitrosomonas europaea. And third one is the Ideonella sacchansi. It is currently tried for recycling of PET plastic. பிளாஸ்டிக்கே யூஸ் பண்ணக்கூடாது அப்படின்னு சொல்கிறாங்க இல்லையா. இப்போ பெட் பிளாஸ்டிக்னு ஒன்று வருது, பெட் பாட்டில்ஸ் we are having, no? Ideonella sacchansis is currently tried for recycling of PET plastic. These bacteria use petase and methatase enzymes to break down this PET plastic into terephthalic acid and ethylene glycol. ஸோ இந்த Ideonella sacchansis இது என்ன பண்ணும் அப்படின்னா பெட் பிளாஸ்டிக்கில் தன்னுடைய வேலையை செய்யும்பொழுது இட் ப்ரொடியூசஸ் an enzyme called PET, petase and MHETase, okay, to break down the PET plastic into terephthalic acid and ethylene glycol. These are the aerobic microbes. Some of the anaerobic microbes degrade the pollutants in the absence of oxygen. So Dichloromonas aromatica has the ability to degrade benzene anaerobically and to oxidize toluene and xylene. Next is the Phanerochaete chrysopodium, an anaerobic fungus, exhibits strong potential for bioremediations of pesticides and polyaromatic hydrocarbons, dyes, trinitrotoluenes, cyanides, carbon tetrachloride, etc. Dihalococcide species are responsible for anaerobic bioremediation of toxic trichloroethene to non-toxic ethane. Trichloroethene என்றது ஒரு டாக்ஸிக்கான சப்ஸ்டன்ஸ் is converted into non-toxic ethane by dihalococoids. So Pestalotiopsis microspora is a species of endophytic fungus capable of breaking down and digesting polyurethane. This makes the fungus a potential candidate for bioremediation projects involving large quantities of plastics. Okay, in this Ideonella sacchansi is a very important question, is a 3-mark question. What is the action of Ideonella? This Ideonella breaks down the PET into terephthalic acid and ethylene glycol by producing an enzyme that is the petase and methatase. It eats the terephthalic acid and ethylene glycol. So it's a very important question. So all the microbes, some are pathogenic and some may have a very good effect on this. Bioremediation involves 2. One is aerobic microbes and another one is anaerobic microbes. Aerobic microbes includes 3. Pseudomonas putida, which is called a superbug. Second one is Nitrosomonas europaea and third one is Ideonella sacchansis. And in anaerobic microbes, first one is the Dichloromonas aromatica, and second one is the Phanerochaete chrysoporium, and third one is the Dihalococcoids, and fourth one is the Pestalotiopsis. So these are used for converting the toxic substances into non-toxic substances. All right. So it's a very simple lesson and a short lesson too. Please go through that. Please go through all the evaluation questions. I'm attaching the PDF to this link. So please go through all the questions and write these questions in your classwork notebook. If you have any doubt, feel free to ask me. Thank you.